Lab 12. Gas exchange in plants. Introduction. Leaves are the primary photosynthetic organs of most plants. Leaf surfaces are equipped with small openings or pores called stomata. Leaf stomata are principal means of gas exchange in plants. The number of stomata on leaf surfaces varies widely among different species of plants. Generally, the lower surface of the leaf tends to have a higher total than the upper. In some leaves, stomata occur in patches. That is, there are areas, usually around veins, that are completely absent of stomata. Botanists have made stomatal counts for many species. Their data indicates that the number of stomata may vary from 0 on the upper epidermis of an apple leaf to as high as 58,140 per square centimeter on the lower epidermis of the black oak leaves. Stomata are opened or closed under the control of a pair of banana-shaped cells called guard cells. When water enters the guard cells, they swell and lengthen. This causes them to move apart and form an opening. When open, stomata allow carbon dioxide, CO2, to enter the leaf for the synthesis of glucose, and also allow for water, H2O, and free oxygen, O2, to escape into the environment. In addition to opening and closing the stomata, plants may exert control over their gas exchange rates by varying stomata density in new leaves when they are produced, such as in the spring or summer. The more stomata per unit area, stomata density, the more carbon dioxide, CO2, can be taken up, and the more water that can be released. Thus, higher stomata density can greatly amplify the potential for behavioral control over water loss rate and CO2 carbon dioxide uptake. But why might it be helpful for a plant to control its rates of water loss and carbon dioxide uptake? One answer can be found in the sun. Generally, photosynthetic cells in plants are only designed to function well over a rather narrow range of temperatures. When overheated, photosynthetic cells begin to malfunction. To avoid this, an individual plant may open its stomata and evaporate water. This will lower the leaf temperature. Thus, one may hypothesize that leaves in the sun should have higher stomata density than do leaves in the shade. But, on the other hand, if water is not available, such as under drought conditions, excessive evaporation might lead to dying out and an equally severe disruption of photosynthetic function. Thus, one might expect plant leaves exposed to drought conditions to have fewer stomata in sunlit environments. The objective of this lab is to determine the stomata density of two different plants. Materials: clear nail polish, clear cellophane tape, microscope slide, and a microscope. Procedure: 1. Leaves from two different plants have been collected. One is labeled specimen 1 and the other is labeled specimen 2. Take a single leaf from each specimen. 2. Using a ruler, measure the length of the leaf from the base of the leaf, not the stem, to the tip of the leaf. Record this information in millimeters on your lab handout. 3. A spot on the top side and underside of each leaf should be painted with clear fingernail polish. A spot approximately the size of a dime or nickel is sufficient to provide enough leaf surface to make the count. Choose a spot that does not contain any of the veins of the leaf. 4. Allow the fingernail polish to dry completely. Then apply a second coat if needed. 5. Firmly press a short strip of clear tape over the dried nail polish on the bottom of the leaf. Carefully peel the tape from the leaf and affix it lengthwise to a clean microscope slide. Use a pen to label the slide with the plant name and bottom. 6. Repeat this process for the top also and for each of the collected leaves. Be sure to label each slide. Organization of data is critical. 7. Observe the casts from the top and bottom of each leaf on high power. Count the number of stomata on each cast. To successfully count the number of stomata in a field of view, you will need to adjust the diaphragm and focus using the fine adjustment up and down to bring different planes into focus. Record your data on Table 1 of your handout. 8. Make a drawing of the top and bottom view of each leaf surface. Be sure to label each drawing accurately. 9. Calculate the area of the field of view in millimeters. Then calculate the stomata density using the formula stomatal density equals the number of stomata divided by the area. 10. Clean up your lab station, clean all slides, and turn off the microscope.